Pharmaceutical Processing, in conjunction with Interfex 2018, presents Interfex Live.
So, welcome back. By far, this title has been the one that we have discussed many, many times. I'm sure a lot of you are here, and it's got a lot of words in it. So it's Emerging and Transformational Technologies in Personalized Medicine. If you sat here for the last day and a half, you've heard that, well, you've heard it here at least 4 times from the previous speakers. So we're going to be coming at this from a slightly different approach. We're going to be looking at it, but what we're going to do, we're going to have our panelists, we're going to level it out so we understand what we're going to have our conversation. You know, in other words, you know, there's a lot of commonality words that we've used. We're going to set it and frame it so that we know the structure in which we're going to discuss it. Right? This is what I think that's the approach we want to take. So, our panelists are, that's Katie, right? And we got Mike and my old friend Mark. We won't tell them how old we are, right?
Thanks,
Russ. So, what we're going to do, Katie, we're going to jump right to you. We're going to talk about the title of the panel is what I had just repeated. Why don't we discuss about emerging technologies and why the adjective transformational has been used? Okay?
All right. Well, first, thank you for your moderation, and I thank my colleagues and esteemed panelists, and we thank each of you for being here. We're excited to share a very new and I'll use the word transformational air purification technology. I'm a clinical provider of in vitro fertilization, and we began our process many years ago. We wanted to remove the variable of air, and we thought we would do so in an ISO 5 cleanroom environment, and we didn't. We had excellent control of our non-viables, but what we learned over a 10-year study was that your low-level viable particulates and your volatile organic compounds, or your chemical pathogens, airborne pathogens, were extremely impactful to our process, extremely impactful to the human embryo, i.e. the living cell. So once we understood and defined the metrics that were truly needed for the living cell, for optimal living cell culture, which exceeded GMP and ISO guidelines, once we learned these metrics that were necessary, we sought to purchase such a system that would deliver the air we now knew was necessary, and there wasn't anything available. So that was the evolution of LifeAire systems. And when we talk about transformational, it's transformational because of the metrics that defined it and the metrics that it delivers. We actually guarantee the air that it delivers below detection VOCs, below detection viables, and ISO 5 non-viables. And also it delivers such at 20 to 25 air changes per hour. So transformational has also been, as a clinical provider, patient care is paramount to me. So the impact by comprehensively removing the variable of air from your process, because everyone is trying their hardest on their protocols, their SOPs, with every step they're taking in cell processing. But by removing the variable of air, VOCs and viables, in addition to your non-viables, the clinical impact that has had on the living cell has been overwhelming.
Okay, so we see this now. Okay, so let's talk about the technology. So we have this unique air purification system, Katie. So how does this differ? And we're going to deal with the other one, the paradigm shift now. So let's talk about that part, okay? The technology, and we'll go with that.
In our clean room, again, we were trying to exceed GMP and ISO metrics. We had 450 pounds of carbon and potassium permanganate as our VOC removal media. We had ALPA filters everywhere you could look, as well as UV. So we very much had a capture model to protect our clinical and laboratory space. So the uniqueness of this technology is that it's a kill model. The technology penetrates the plasma wall of your cells, bacteria, viruses, and destroys the RNA and DNA. I did not want a capture model involved in this technology. It's also, what makes it unique is the engineered molecular media. there's no one fix. So carbon is going to adsorb certain biochemical families of VOCs. Potassium permanganate is going to oxidize others. But there are many cytotoxic, highly cytotoxic VOCs that go right through both media. So our media is targeted for all cytotoxic VOCs, but it's done so in 5 different stages.
So we'll talk about the stages in a bit, but I mean, essentially what we're talking about is a shift here where we actually have a clinical outcome. Mm-hmm. All right, maybe we should just, so we got that part clear. It was a viable living cell. We had actual clinical data that for the personalized medicine aspect. Right?
Yes, our system, I mean, this is a new technology, so we've just recently commercially launched, but Stanford, Mayo Clinic, Northwestern, Wake Forest, Iowa, UConn, and others have been using the technology, and just by comprehensively removing that variable beyond your non-viables, beyond GMP guidelines, and they're all they're processing differently. Their clinical outcomes have increased by 20 percentage points, which is about a 40 to 50% increase.
Okay, so that's the background. Now we're going to draw it into what we call the bio industry or biotech, where you would use this. Okay, so to frame that out, we'll talk. So why don't we, again, Kate, I'm going to look to you. So where would we frame this out in the biopharma and personalized medicine area? Then we'll go on to facilities and how we implement it and how we start to bring it to what we understand. Okay.
The human embryo is one of your most sophisticated living cells, but it's really identical to the cells with which you're working. Any living cell application, gene cell therapy, regenerative medicine, stem cell, the cells we worked with, as yours, have zero defense mechanisms, zero, to their environment. Your volatile organic compounds, again, I'm just trying to share pearls of what we learned the hard way, each have a partition coefficient. So they're either water soluble, oil soluble, or often biphasic, which means they're going to enter your growth media that's supporting your cells in a fraction of a second, or your oil overlay, or they can actually reside in both. And once they're present, it's a permanent threat. They're not going to go anywhere. They're highly cytotoxic. They actually influence the epigenetic variation, the DNA methylation of those cells.
Okay, so we're talking about material which is in the environment. We hadn't accounted for it before. This technology now deals with that. So what I'm going to do, we're going to start to work back into the facility aspect. So Mark, what's the biggest impact on this shift to the personalized aspect and facility design? And then Mike, we're going to get back to, implementation that as well. So why don't we start from, I always try to explain to the group, I always talk about the box everything fits in, and then we talk about the box inside the box. So
great,
let's go with that.
All right, Russ, so thanks. So, there's really 2 types of cell therapy. So you have autologous therapies, and then you have allogeneic therapies. So in the autologous therapies, those are, you take cells from donors, then you process them in a clean room environment. You alter them in some way, you separate the stem cells, and you find the product that you're really looking to put back inside the patient. So that's one shift. On the allogeneic side, you're really looking at donors that are going to go into multiple people. So as this personalized medicine starts to evolve, especially in the autologous example, you're not making a large batch as we're used to in a large batch process. You have a bag of blood from a donor who may be terminal cancer, right? And you're trying to find that cure. So now you start to process those very precious cells, hoping to be able to cure that person, or when they're done their chemotherapy regimen, actually regenerate bone marrow and those kind of things. So the shift now becomes, again, very personalized. Now what that does is, instead of designing facilities that are larger and designed for a large multi-product facility or a single product facility, you now have to start with every patient is a different batch, and again, it's very precious. So these facilities are getting much smaller. They're starting to look like a clean room lab environment attached to maybe a blood treatment facility. They're close to hospitals and cancer treatment centers, and they look very differently to the GMP environments that we're used to seeing when we walk around facilities like you're seeing equipment going into large facilities today. Mm-hmm. So you really need to start looking at all of the impacts to the product. Now, there's new technologies that will do this benchtop processing in a closed environment, but there's so many different manipulations to those cells that at some point you're in a biosafety cabinet and you're taking air from the room, which might be HEPA filtered, but you're not taking out or knocking out the VOCs, which is what Katie's product does. It really starts to improve that environment. So from that facility side, we start to scale down, mm-hmm. and we start to build these smaller, flexible facilities, and they fit very nicely into a pod environment, which
right.
which Maik produces.
So Maik, that's where so you see the flow I'm gotten here. So what I want to talk, what are the biggest, again, same question, a little different. Biggest differences in processing of personalized medicinal products as opposed to what we're used to seeing. In this case, this is where it happens. So from a pod design, what are we talking?
Yeah, ultimately, when you look at personalized medicine, number one for me, it's the platinum standard of aseptic processing. So you cannot sterilize, in instances, some of the product. And then again, to Mark's point, it's a very small volume. It's a patient. You're dealing with a patient. And when you're dealing with a patient, you need to make sure that you have the most robust containment from the apheresis process, for example, all the way to the reprocessing of the sample and growing the cells. So you don't want to have any influences, negative influences, either by microbial ingress, which is always like a major focus, but also volatiles. And a good example, there are two good examples in regard to influences of volatiles. The one is the leachable or leachables, which come out of single-use process technology. When you actually have a small volume, you can have leachables coming out of single-use process technology. You have to be very aware about that and avoid these type of leachables. We've seen incidences where CHO cell growth is influenced by leachables. Now, when it comes to volatiles in the air, which is another component, which I have to say I never thought about, and then we talked with Katie and then started really thinking about it. Volatiles are in the air all over. They can be brought in by the human being. They can be brought in by clean room materials, for example, the wall panels, the laminate floor, for example. So we have to be much more aware about that, because these volatiles penetrate HEPA filters, which are installed in isolators, and therefore we require to clean the air within a confined, smaller compact clean room system. And we can do that with a LifeAire system. And then when the air is cleaned within that compact system, we can process samples within an isolator. And another good example, one of our companies, my boss's company, is actually, they utilize cancer cells from patients, extract the cancer cells, bring them into an isolator, and reconstitute them, highlight them. So your own body is fighting the cancer, right? So it's a cancer vaccine product. Now, this is an open process. If you do not have the LifeAire system and bring the volatiles out, these systems, these cells will be influenced. And you don't want to have that happen. So it's very important that the containment of the samples, the processing of the sample, as well as the air within that containment has to be assured clean.
I think the thing that really struck me, Katie and I had an opportunity to talk about a month ago, and we were talking about raw data, which we're not going to talk about. But anyway, there was a blip, and she was explaining some, was it Penn State? Was it? Where was that?
Lehigh Valley Hospital.
All right. But okay, so, and there was a VOC blip, and what you finally tracked it down to what?
So we, in our ISO 5 clean room, began out of the gate. Now this was 18 years ago. So the national clinical pregnancy rate in the United States was about 30%. So we began out of the gate at about 50%, and we were doing cartwheels down the hall. We thought, this is it. Everyone should do an IVF in an ISO 5 clean room. And then we repeated that a second time with another 50 to 60 couples. So we thought, all right, this is not an anomaly, this is real. The third cycle, because you coordinate your patients to ovulate, the third cycle, we dropped to 13%. And that's what he's referring to. So imagine, put yourself in our shoes. You've counseled a couple in May, and your pregnancy rate is 52%, or your living cell success rate is X. But by the time they come through your process, you're at 13%. But the worst is that you don't know why. So part of our learning of the significance and what level VOCs we truly needed to have beyond what our clean room was providing. I was leaving the hospital that night, or one evening, and I noticed we're a level 1 trauma center. So I noticed they were resurfacing the medevac pad, and I could see the fumes coming up off the asphalt. That's toluene. So I asked him for the MSDS sheet. I asked him when they started the process and when they're going to finish the process. And then we went back and looked at our live VOC data that we were collecting, and we had parts per billion levels of toluene, which was coincident with the epigenetic variation that we then later saw in our cells and the drop in pregnancy rate.
And these sorts of aberrant behavior you've seen.
Yes, we have 10 years of data linking environmental incidents, common. None of us can control what's going to happen outside of our space. Not only outside, but what events were occurring within the laboratory or by the staff, a protocol change. I mean, subtle things that are going to happen. We were able to correlate environmental activities with breaches of our clean room. Low-level VOCs, low-level viables, and then the clinical impact or the impact on the living cell that it had.
Right. I think the one that really struck me was the excursion you've seen, and you linked it back to, as I recall, it was a homecoming.
Yeah, oh yes,
homecoming.
Tailgating. Yes.
That's pretty good.
That's why you said Penn State.
Yeah, right.
Yes,
I couldn't remember which one it was.
It was actually University of Iowa.
Okay.
Yes, their pregnancy rates would drop, and it took them years to figure this out. They now have our system, so tailgating is no longer an issue. But they could not figure out why their clinical outcomes were dropping only during football season. And then once they put our system in, and we do a lot of air testing before, after we test the molecular media at 9 months, we can tell which VOCs have challenged that particular location. And that often helps them go back and revisit and improve their own SOPs, because we're telling them what's in the space that's cytotoxic to their process. But then once they put our system in and they had additional data, they realized it was the fumes from the tailgating at the football games that were creating these additional VOCs and impacting their cells.
Okay. So let's get back to Maik. Now, I think what I wanted to talk is, I found this incredibly fascinating. However, I kind of got hung up on it unnecessarily. But I want to get back to the ground. So what are the critical success factors from somebody who's an expert in the industry? We're talking essentially about this shift to personalized medicine. What are the key success factors?
Very good question. Number one, I would like to add to this, because we always think, when it comes to single-use process technology within that personalized medicine field, we always think about the microbial barrier. But then there's no barrier for these volatiles. So that's one part which I would just like to emphasize. We always think microbial, so do the regulators. And it's not about restricted to IVF, it's specifically also important for personalized medicine. So success factors which I see for when we installed actually cell and gene therapy platforms with our pods, clean room pods. And the success factor there is speed, number one. We need to be very fast nowadays with building, processing and manufacturing spaces, because once you hit the target, once you see the success of the target, there's a sense of urgency. We need to get clinical material as fast as possible. Very good example is Avexis. We actually supplied a GCon pod cluster to manufacture clinical material. It's a pediatric gene therapy. So for us, it was very important to be fast, have this facility up and running in months. So that's one part. The other part is also, which we see very often, is scaling. So you built your site and you work this site, and now you suddenly realize, I need to scale up. What do I do with scaling up? Typically when you scale up and your infrastructure is interconnected, your HVAC system is interconnected, when you scale up, when you break the wall and you want to build another processing space, you start interrupting the existing site. And that's the reason to the point of LifeAire and POTS, and also the work together which we do with IPS to create independent, autonomous clean room spaces, which are highly containment units, but also can be still run when you dock an additional space into that room. So when you start scaling out your capacity, you don't interrupt your existing processes. So very important, high containment, speed, absolutely of essence, and then scalability. The other part which I always like to see is also clone it. When you have the capability to clone it and bring it from, you standardize on one particular clean room structure, then don't invent the wheel over and over again. Clone it, copy it, you get scaling effects, you will be much faster with your delivery in future.
To me, scale out is way better than scale up.
Yep.
Right? Anybody that comes to me when I do consulting work, my answer is, well, just make another one.
Yep.
If 700's good, make another 700.
Yep.
But let's reel back on the personalized medicine. So what we've heard is, speed to market, personalized medicine, artifacts that we didn't anticipate coming from the environment, things giving us trouble that, oh, by the way, there's no regs around, All right, okay, I think I get that. So what are we talking? So it's a paradigm shift, it's personalized medicine, it's live cells. So we got the pod. So LifeAire, the system we're talking about, so that gets, why don't we talk about what that is, and that gets bolted on or in, and then we'll go back. Now that we had the ground leveled out, why don't we talk about what's so unique, how it works, and where we're going, okay?
Well, this system replaces about 13 feet of the ductwork, and we prefer to put it closest to the space that you're trying to protect. It's a multi-staged system. Numerous stages, again, engineered molecular media to take your VOCs to below detection, and that's all cytotoxic VOCs down to below detection, critical for the process. We deliver non-viables at an ISO 5 level, and then the viables we take. The system was, because it was designed to protect the human embryo, we chose the most difficult airborne pathogens for it to remediate. So on the biological side, we chose the anthrax spore, because if you can kill the anthrax spore, you'll provide a 9-log reduction against the things that are more impactful to us. So the system delivers air that's below detection in viables, below detection in total VOCs, and ISO 5 non-viables. We actually guarantee the air that comes out of the system. And again, it replaces 13 feet of ductwork downstream of your air handler and conditioned air, and closest to your final diffuser as possible.
So Katie, why don't we talk about how fast is a system like this? I mean, we talk about something that takes, because Mike just talked about, we got to get there and we got to get there fast. We got to get into the clinic. And remember, the trip from clinic to product is pretty short when we're talking about this stuff. So let's just say, I have this revelation that I need it. How fast does this thing get turned around, fabrication-wise, for the system? I'll get to you, Mike, and we'll get back and then we'll get to the facility.
Sure.
Let's start here. Go.
The systems can be manufactured in about 4 weeks.
Wow. Okay, so Mike, to put them into a pod, how long does that take?
Right now, when we talk about one mini pod, we put the LifeAire system in it, we can deliver that pod in 3 months. So then we do the FAT now. It's a prefabricated clean room system which we build. So we put the LifeAire system into our pod structure, prefabricate the unit in our facility, We then do factory acceptances. So 3 months to factory acceptance test, then we ship the unit wherever it goes. My vision though is, and I know some people say this guy must be crazy, but ultimately when we actually start standardizing on these kind of structures, these are mini clean room, prefabricated mini clean room structures, I don't see any reason why it should be more complex than a car. So when you go in a car showroom right now, in 6 weeks, or even less, you get your car reconfigured how you want to have it. I don't see any reason why we should not be able to do the same thing with prefabricated clean room systems. So you standardize your clean room, put the LifeAire system in it, and then you tell us, okay, what automation platform do you want to have it in it? That's the configuration, and then we deliver the unit within weeks.
Okay, so where I've always been, and it's where Mark and I work together, is that's to me ideal. So if you got a warehouse, a garage, you tell me, I back this thing in, whammo, I'm in business. So you take this into third world, you take this to where there's critical need, government, it's agencies, and you start to create the most critical live vaccines. But I think sometimes I overstate it. So Mark, the garage or the warehouse, maybe not. So why don't we talk about that a little bit?
Well, I'm glad you brought that up, because just like you've got a very quick filter fabrication process and you've got a pod process, what we've been doing with GCon is pre-engineering these, so investing the time now so that they are pre-engineered and standard, so that a standard design can happen very quickly. Now, usually owners will want to customize things, and they'll want to, again, customize different things and fit them to the utilities that they have in place versus buying maybe some redundant systems, because the pods are shipped fully self-contained. They can just basically wheel them in, move them around on an air cushion, and then plug them in with power to get them running. So in a standard facility design, it usually takes a long time. We're talking 3 months to design a concept, a couple of months for basis of design, and then the detailed design could take some time. So the paradigm shift here, being driven by patients' needs and a need to get to the market quick, is to do some of this pre-engineered up front. The technology advances for single-use technology, plug and play, and all of the things that we're starting to see vendors produce become very easy to take that platform, put it inside the box, clean the air up, and then ship the product very quickly. So it is definitely a paradigm shift.
Absolutely, and we've been talking about it. Bottom line is, you go from no facility in country to facility in country, as long as I got the spot. Back it up into the Star Lifter and I'm going.
Absolutely.
All right, so.
We've actually taken it one step further and created a product called Icon, which is a joint venture between IPS and GCon, and we've brought Butler Span into the mix, so they can actually build a prefabricated box, and then you put the pods in it. So it's a box in a box concept, that there are base models that very flexible for both personalized medicine as well as normal biomanufacturing.
Right, I think that's the most important. I think that's one of the key pieces that we have. So Katie, we're talking personalized medicine, but what this does is it's in our space in biotech. It's right there. In fact, it's beyond what we would need. So the answer is to deal with facility shortcomings, lack of capacity. This stuff has got it all written all over it. Okay, now, having said that, let's rewind. So Katie, if this thing is as great as it is, what's the cost factor? People are going to want to know what's the return on an investment. Is this thing going to go like, we used the car example, so we go from a Chevy to a Rolls Royce, or what do we do? What are we talking about? What would be give us a sense.
Well, there are different size systems of the LifeAire technology, and it's all based on cubic volume of air, the air that you're trying to protect. So that's the first calculation that's done, and then the system is sized, and then our engineers can look to see where it can be best placed. But it's a one-time capital cost. As far as maintenance, there really isn't maintenance. It's a passive system. There are no moving parts. We recommend in the first 9 months that everything is changed out. Again, you've got to be proactive, whether it's in vitro fertilization, healthcare, gene cell therapy. These cells have an exceptionally high therapeutic value and life potential value. So time is of essence. You have to be proactive with this. So we recommend changing out all of the internal components, the filtration components at a 9-month cadence. And then actually we analyze the molecular media that comes out of the system because your use here in New York, you're going to be exposed to different airborne pathogens than you would in Pennsylvania, than you would in California, not only environmentally, but from the protocols or the application that you're using. So we assess for not only life left, but what volatiles have been hitting our molecular media, what our system has been challenged by, and then we can tell them exactly what's been in their environment, and often they've used that to improve their SOPs. So cost is the initial capital expense, which again is based on the cubic volume of air. Maintenance is 0, replacements is every 9 to 12 months. Return on investment, improve cell viability, reduce loss, reduce contamination. I mean, we've seen the impact on both the positive side and on the loss side. Loss reduction of loss, improve viability.
So you pull the media out, you test it. So what is that? You come back and you, with the client or with the user, you come up with a recycling. How's that work?
Nothing's recycled because.
Well, see, I meant recalibration.
We'll change everything out at 9 months and then test what the molecular media that's been in their system for their application. And as a provider, when we were ordering and placing equipment for our purposes, we were told change this at 6 months, change this at a year. I never knew, one, was that adequate for our practice, for our patients, for our process, and two, I had a budget. So my goal with LifeAire systems was to give them a data-driven decision on when they should replace all of their components based upon their area and their use.
So it could be that 9 months, the data comes back and tells me I don't need it for 18 months.
We haven't had that happen yet, but it could.
Yeah, or worse yet, it come back and tell me it's got to be a month.
Right. We haven't had that happen either.
Thank God for that, right? Yes. Well, not too many people living on top of the tarmac to tell you when. That's good. Or they're having a tailgate down the street. So, Maik, let me ask you this. So, pulling this stuff out, I mean, let's talk about change out. I mean, what did you have to do to pods, or is this something amenable? I mean, how big a deal is this switching out of the media from your standpoint?
Well, it's not. It's like when it's a typical clean room maintenance setup, which you have with our clean rooms, same thing. So you would, when you utilize the HEPA filters, you would actually change the HEPA filters. When you use the LifeAire filter system, we would do the 9 months maintenance standard as usual pods, as you do it with typical traditional clean room systems. So it's a typical maintenance, either a maintenance shutdown, or you can do it when you actually go not within the clean room space, you go on top of the clean room space. It's a typical maintenance.
Gotcha. So that's stuff you can build in up front, Mark. Is that the way that works?
Oh, absolutely. So, like we would design, a bag in, bag out filter with access space, and that could be just built right in. The really only impact might be, you have HEPA filters, so you have to make sure you have enough static pressure in your fans to overcome the pressure drop of the unit, and they're all sized that way. They tell you, based on the flow, what the pressure drop will be, so it can, be retrofitted into an existing situation or designed in new into a brand new situation.
Yeah, and I think because one of the things about flexibility is how easy is that to do. So, Maik, what I'm hearing from you, it's not a big deal.
No, it's a typical maintenance. It's a very typical maintenance process. It's not a big deal. The one thing what we like with the LifeAire system is extremely compact for us to build into the pod structure. So when you actually have that compact type of system, we can put it into our tech space. So we have a clean room space, we have a gowning space, and we have a tech space. The tech space is totally separated from the clean room space. So when we have that tech space, we can actually run it. The tech space is connected to the gray space. You can just go into our tech space without even touching the clean room space.
Okay, so let's circle back, and I know if I was sitting in the audience, I'd want, okay, I'm trying to weigh, this versus that. So the answer is now we got LifeAire sitting there, we got a current technologies, Mike? And this is what you do. So talk to me. I mean, what's the advantages versus what we do? I mean, we've talked about it, but can you? What do you think? Can you summarize that? Or?
Yeah, it's. I mean.
I'm not trying to put you on the spot.
No, you don't.
Good.
So I'm over 30 years in the industry, and when I look at what we build, right, what we build traditionally, fixed build, one product life cycle, complex, and all interconnected. So what you do is you create a large space, which in instances is difficult to control. So now you go from that traditional build, we see process intensification happening more and more, not only in the personalized and the cell therapy area, but also in the bioprocess area. So these systems become more smaller, more process intensified, which gives us the opportunity to bring our clean room spaces, our clean room infrastructure, and shrink it also down. So when you start shrinking this footprint and the footprint of the critical clean room space, you start reducing your energy cost, you start reducing your running costs. When you then also take the interconnection, the mezzanine space out, and you don't interconnect any longer, but you create like autonomous clean room systems, potentially what you could do is you could capacity flex. So you say, one clean room space right now, this area I don't need, shut it down, seal it off. It doesn't influence my other clean room space. And when I want to have this clean room space up and running again, I VHP it and sanitize it and run it up again. So you create a higher flexibility, you create a lower cost of investment, and you create a lower operating cost because your critical clean room space is much smaller than it was in the past. Plus, the one part what I always see is also when you start looking at these forests of ductwork, you have a leak rate, a certain leak rate within these interconnected systems. With compact systems, you avoid also this type of leak rate.
So Mark, let me just say, Mike, from a facility and critical utility standpoint, I mean, if we decide to go with the LifeAire, which seems to, if that was the way I was going to go, and we hear it's a passive system, so does that impact critical utilities or what?
So it's actually a real synergistic product to put in. So as Mike was saying, in the pod environment, they actually have the tech space. So in a stick-built environment, you'd want to know if you had a walkable ceiling above your clean room, and you just have to build in, the 13-foot plus a little pressure drop, and get it as close to the end point as you want. If you don't have a walkable ceiling, you have to make some maintenance access provisions for that. So maybe you're running ductwork in the corridor and you've got the LifeAire system above the doors outside the clean room, so you can get above the ceiling and do what you need to do. But it's it's a very easy product to integrate into your design. And to kind of reiterate what the purpose here is that removing VOCs in a GMP environment has never really been on the radar. We've always been dealing with viables, non-viables, the particulates, and we certify clean rooms from when we do our GMPs and validations, and we're looking at particulate, right? And the VOCs are getting through the HEPAs. So it's not until you start to see the impact on the in vitro fertilization field that you start to say, why are some of our open processes, cell therapy, why are we losing batches? And people don't know that. So right now, at CHOP and the University of Penn and all these other places that are starting to do this, again, with patient samples that are very limited in terms of that resource, and they lose a sample, they're trying to figure out, why did we do that? Did we do something wrong? Did we contaminate the cells? How did we do it? And yet it could be something as simple as the isopropyl alcohol or other things that are in the facility that are getting through the HEPAs and are actually killing the cells. So again, this is a product that when you do a risk assessment, you take a look at it, and it's something that really can enhance the viability of your processes.
All right, so let me ask you the question I'm sure is on everybody else's mind. So what we're talking about is capturing and mitigating something that we don't normally measure, okay? See, Mike's he's smiling because you know where it's going, right? So I've made this great discovery. So where do you see this thing landing in the requirements and the regulations then? I mean, I'm speculative, but if nobody's thinking about it,
Can I take a shot at this?
Sure.
Either way, let's talk.
About 10 years ago, the Green Building Institute started to get together, and we started to LEED certify buildings. And what do? We started purging buildings of the VOCs before we put people inside. To get your LEED certification, you're off-gassing from carpets, you're off-gassing from the furniture and those sort of things. So we started to do that. And that's now built into a lot of the building and energy codes, but the reality is you're creating a cleaner environment. So as we start to look at cell processing, this is where it gets pretty exciting, because you have success rates that you can quantify, and then you also know where things are failing. So to me, it's almost a natural to start to monitor this and to say we're going to mitigate this and design it in as a risk prevention process.
I wanted to go back to a point that you were making, Mark. It was not only the identification that these VOCs were so cytotoxic, but it was the level at which they were, at parts per billion. So our space was doing a good job controlling them at parts per million, but that's log levels higher than what the cells truly needed. And to your question, we've actually submitted our technology to the FDA Emerging Technology team for their awareness and involvement.
Okay, we got Mike, I'm going to throw it open to questions unless you got a final comment.
Yeah, just to that point, I'm the chair of the Science Advisory Board at PDA. We actually have discussions about not just about the volatiles in the air, but also about the leachable impact on the clinical. And so I think the discussion is becoming more and more prevalent, and specifically due to the fact that we have such a small sample size. We cannot lose that. We cannot lose that patient sample. So I think it becomes a topic which we most definitely need to address and attack. I mean, we don't want to have new car smell in our clean room areas. We need to check the materials which we utilize within the clean room spaces, the process equipment which we utilize in the clean room spaces, and what can we do about it in regard to filtering out volatiles.
Okay, what I suggest you do is you tackle these three on their way out. We got to get ready for the next panel though. Thank you.
Thank you.
Thank you.